present today the final molecular genetic findings using phenotypic and sequencing data of the Translate NAMSA project. Many of you are already familiar with the project that was a novel concept of care of the patients with suspected rare disease that was implemented and evaluated in a 3-year prospective study in Germany. Generally speaking, rare diseases are not that rare as one might think, especially if one asks human geneticists. According to the recent analysis of the OFANA database, rare diseases affect up to 6% of the population. At a maximum prevalence of 1 to 50,000, the diseases defined as ultra-rare. And this was basically the focus of our study, since many of the patients included had previously received different types of genetic testing, such as RACGH or single gene sequencing. There are many challenges in the care of patients with rare diseases, particularly the severely delayed time to diagnosis. On average, it takes about 4 years. Furthermore, many patients require multidisciplinary care and comprehensive genetic testing, and therefore, one of the goals of the project was to implement exome sequencing in a standard healthcare. Study participants were enrolled from 10 different centers. The exome sequencing was performed by 4 institutes of human genetics in Bonn, Tübingen, Munich, and Berlin. The project was funded by Gemeinsames Bundesausschuss and supported by healthcare insurance companies and AXA. Professor Krude will present tomorrow the final results of the evaluation of the project. So between January 2018 and December 2020, in total 5,652 patients were enrolled in the study. Patients with a likely genetic cause were comprehensively assessed by multidisciplinary teams with domain relevant clinical and genetic expertise. For part of the patient, additional tests were recommended, and consequently, exome sequencing was initiated for 1,309 pediatric patients and 268 adults. The sequencing results were again discussed in a case conference, which either confirmed the diagnosis or detection of a variant of unknown significance, or maybe a candidate gene that was further investigated on a research basis. Conclusive monogenic diagnoses were established in 497 individuals, of which 400 were associated with unique diagnostic rate genes, suggesting again that ultra-rare disorders were enriched in the TN cohort. In addition, we described 56 novel gene phenotype association, and within the study, 28 genes subsequently reached the VGD status or a high evidence score. Concerning the leading clinical symptom, each patient was assigned to one of 6 disease groups. The neurodevelopmental group yielded a particularly high proportion of novel disease genes. The diagnostic yield was 5 percentage points higher for children than for adults. And this can be explained exactly by the fact that the exome sequencing in children was primarily like the indication for exome sequencing in children were primarily neurodevelopmental disorders, whereas neurological or neuromuscular disorders were the predominant indication in adults. We analyzed the diagnostic yield not only per disease group, but also based on the phenotypic features, using terminology of the human phenotype ontology. On average, 5 HPO terms were assigned per individual. In the next step, we analyzed the association of the HPO terms with the diagnostic yield using least absolute shrinkage and selection operator analysis. So the coefficient paths of regression analysis will be shown on the right. The more to the left the coefficient path starts to deviate from the x-axis, the more informative is the term for predicting the diagnostic yield. So features with positive coefficients were associated with increased diagnostic yield, and in contrast, features with negative coefficients seemed less likely for a monogenic cause. So the lasso analysis yielded dysfunction of higher cognitive abilities, abnormality of the limbs, and ataxia as predictive clinical features. This model was validated on an independent cohort and is comparable and is available as a web service that can be used to estimate the diagnostic yield given the phenotypic features of the patient. number of 211 individuals gave consent to a computer-assisted prioritization of exome data by image analysis. And yeah, the DIA study that was an associated research project. In 88 patients, the diagnosis could be established by exome sequencing. And in 48 out of these 88 cases, the underlying disease is supported by artificial intelligence deep gestalt. That means that the phenotype of the disorder has been learned by the artificial intelligence in advance, and the facial gestalt of the patient can be evaluated based on the portrait image. On the right, you see a graphic that reminds of the first publication of the PADIA study and represents the prospective results in our solved cohort. So the exome variants are ordered according to different scoring approaches, solely to molecular deleteriousness, CAT score, to a score from facial gestalt, the deep gestalt score, and the PADIA score that includes them both, and a clinical feature-based semantic similarity score. So the area under the curve is the largest for PADIA scoring. And this means that this approach not only speeds up data analysis, but yields additional evidence for variant classification, particularly if the gestalt is quantified as highly similar in a phenotype of high distinctiveness. This pie chart shows the modes of inheritance in the solved cases. So disease-causing variants most frequently occurred de novo. Regarding the autosomal recessive disorders, we also checked whether the diagnosis or the higher risk for the diagnosis could be established by extended carrier screening. And yes, at least in 73% of all cases, the higher risk for the disease could have been identified. This bar graph shows number of variants relative to the time interval in which the gene was first described as a disease gene. And we see that the variants reported in the TM cohort were found mostly in genes that have only recently been associated with diseases. In contrast to other larger cohort, like from NHS England, most diagnoses established in translate NAMSA are ultra-rare, and most frequent diagnoses were simply underrepresented. Regarding the novel disease candidate genes, we defined following mandatory criteria. So the gene was not previously associated with human phenotype. There was no other explanation found, and the variant was absent in controls or co-segregated with the disease. We defined also characteristics such as nomad constraint metrics or functional data by which the level of evidence of the candidates were assessed. Finally, I would like to briefly present you a few case reports of particular interest. So here we see an example of novel gene phenotype association that could be established in translate NAMSA. So the patient presented developmental delay, hypoplasia of corpus callosum, dysmorphic facial features, and a distinctive type of synpolydactyly. Exome sequencing revealed biallelic loss of function variants in MAPCAP5, encoding the mitogen activated protein kinase. The functional analysis showed no expression of MAPCAP5 protein isoforms and reduced levels of MAPCAP5 interacting protein in patient-derived cells. Here we see another very interesting case where computer-assisted image analysis led to diagnosis. So the female patient presented developmental delay, moderate intellectual disability, and distinct dysmorphic facial features such as upslating palpebral fissures or prominent bulbous nasal tip. So the computer-assisted image analysis revealed highly similarity to a specific syndrome. The initially negative exome data were reanalyzed under consideration of this aspect, and complementary genome sequencing was performed and showed intragenic deletion associated with Cullen-de Vries syndrome. For 18 cases classified as uncertain or unsolved, multi-omics assays such as metallome, transcriptome, or proteome analysis were initiated. A very interesting approach of methylation analysis could be performed in a patient with de novo variant in KMT D, and epigenetic signatures help to clarify the status of this variant as rather likely to be benign using support vector machine probability scores. We're particularly happy and satisfied with 5 diagnosed patients of whom personalized treatments or therapies directed against the mechanism of the disease could be initiated or recommended. Of note is that the gene therapeutic approach for the metachromatic leukodystrophy was authorized in December 2020. I would like to emphasize one case, one patient. So you will see this 9-year female patient showed developmental delay, intellectual disability, and autism spectrum disorder. The brain MRI revealed delayed myelination and deficits of the white matter. So the exome sequencing showed a missense mutation associated with creatine deficiency syndrome 1. And during the following 18 months after the treatment initiation, she showed improvement in her motor function, social behavior, and weight gain. And this is exactly what we want to see and hopefully will see more in the future. So, just a few words in conclusion. So first of all, thank you for your attention. It's nearly impossible to name everyone who worked on this project. So I would just like to say thank you to Professor Heiko Kruder for his great encouragement as the head of Translate NAMSA Consortium, and to Professor Peter Kravitz, who coordinated and supervised our working group responsible for the analysis presented today. And again, thank you to all of you who worked and supported this project. Important work for rare diseases in Germany is done, and yeah, what can I say? To be continued. Thank you.
Any questions?
Entertainer-Qualität. Sehr gerne. Ja. Gut.
I would be interested in the classification of the phenotypes. You showed that there were 6 groups, but there were a lot of patients with syndromic features. So were there any other criteria how you categorize these?
Yeah, thank you for this question. So I'll just take here we see on the slide distribution of the phenotypes in the TN cohort. So basically the phenotypic space in the background was, you don't see it actually, or maybe we can, I don't know if we can show the one slide for this, to answer this question, please. Oh, thank you so much. Yeah, because I think it's a very good graphic to describe, to answer your question. So we see here the distribution of all the phenotypes in the TN cohort and phenotypic space in the background. So those gray dots in the backgrounds is defined as by all diagnoses according to their HPO annotations, and all the patients are color-coded by the regarding the disease category. And here we see also patients who contributed to identification of novel gene phenotypes associations. So we see that the cohort is actually representative for all rare diseases, in fact, and we also see here a good overlap between the neurodevelopmental group and neurological group. So in fact, there was no like criteria which patients could be enrolled in the study, but every patient, I mean, with suspected rare disease and not confirmed diagnosis.
Thank you, Magda. Great talk. You nicely mentioned that artificial intelligence actually does help to diagnose patients. My question is, if you compare now single exome versus trio exome versus then using artificial intelligence, does that influence the diagnostic yields at all? basically, to say it really hardcore, does trio exome sequencing give you the best diagnostic yields and you don't need any artificial intelligence?
Well, we had with so a nice example where actually the artificial intelligence helped to diagnose the patient, because in fact the deletion couldn't be identified by exome sequencing, and in fact the majority of cases received trio exomes in our cohort. So I would still recommend to use the artificial intelligence approach to improve the diagnostic yield, and in fact that's actually what we planned for the unsolved cohort. So first we want to systematically reanalyze the data of unsolved patients and also improve it by artificial intelligence approach and other strategies like genome sequencing or multiomics. Yeah, but it's good to do it. Yeah.
One more question. Yeah.
Yes, thank you very much for the data. If I calculate you have that correct, it's the success rate for really treatment is something like 0.1%, 5 out of 5,000. And the aim of transcendental is actually to speed up treatment or cure of people with rare disease. And in fact, I mean, an increase in weight percentile from 4th percentile to 7th percentile is not really statistically significant. Also, GLUT1 should be contained in a proper neurologic workup. So I wonder, weren't you disappointed just to find a handful of curable diseases? I would have imagined it's like 20%.
No, we were not disappointed.
Well, I would, because, I mean, less than 1%? Are we so bad in medicine?
Pedro, do you want to answer, or should I?
Yeah.
Maybe a short answer. You can discuss it first with you. We get together.
Okay, we know that the number of therapies will increase by 5 per year, yeah? And much harder to establish is the diagnostic pathway. That was our job, and that's what we did.